All right, thank you everybody for joining us this morning. I'm really pleased to introduce our guest speaker today. It's Pat Soranno, or I guess Dr. Patricia Soranno to make it sound really official, from Michigan State University. Here at GLERL, we study principally 5 different lakes, but they're really, really big lakes. And truth be known, we focus our effort on only 3 of them for the most part. In contrast, Pat studies really, really small lakes, but her current database that she has spent some time over the last few years assembling contains over 50,000 of them. So she is able to make inference at spatial and temporal scales that we can't really think about with respect to the Great Lakes. Pat did her PhD at the Center for Limnology in Madison, Wisconsin, and that's when we first met. She is the founding editor of the relatively new journal, Limnology and Oceanography Letters, and I was really pleased when she agreed to come down and talk to us today about some of her current work. And I'll turn it over to you then. Thanks, Pat.
Thanks, Craig. Thank you. So as Craig said, I'm going to talk about every lake except the Great Lakes today, so I apologize in advance. I'm going to talk about a program called Lagos that I've been a part of for the last 6 or 7 years. And since starting that project, I often get asked, well, what have you learned after all of this? This is my attempt to answer that question. And it's the first time that I've tried to pull everything together. So it might be a little rough, and I'm looking for feedback and questions, so feel free to ask the hard questions. Now, when I think about trying to understand lakes at really broad scales, the first thing that comes to mind for me is thinking about variation. And variation in ecology has been something that ecologists have been interested in for some time, from the variation that happens within a lake, such as within an individual population and the body size, such as a great example here of the Atlantic silversides, to variation in the number of species along latitudinal gradients at the global scale. Ecologists have also recognized that variation is strongly tied to the spatial and temporal scales that you're looking at. And I really love this first space-time graph that was created by Henry Stommel in 1963. He was just describing the physical characteristics of the ocean. And after this, especially in the 70s, these Stommel diagrams were used a lot to apply them to ecological processes, recognizing that a variety of different sources of variation vary along space and time gradients. There, in fact, was a lot of research that has been done in the 70s and 80s and well into the 90s that really focused on scale and variation. And I tried to summarize all of this research into 2 either propositions or assumptions that we have about variation in ecology. And the first is that spatial and temporal variation of ecological properties depend on the scale of observation. I don't think anyone would disagree with this. It sounds almost like we've known this forever. And the second proposition is that the dominant controls of variation varies by the grain and the extent of the study area. So these are very general properties about scale and ecological variation. And what we've tried to do in our work is try to dig a little bit deeper, and can we say things about variation and scale in a little more detail than what we've got in these 2 general propositions. So what I'd like to talk about, or start by talking about, is when you dig down a little bit deeper, there's some gaps in our understanding about ecological variation. The first is a lot of what we know about space and time sometimes have happened by synthesizing across very different studies, because conducting a study across broad gradients of space and time takes a lot of data, which we haven't always had. A second important gap is in the general scale of regional to continental ecological processes. A lot of times, so ecologists, we're used to studying at the very fine scale. We also are pretty good at the global scale. but this intermediate regional to continental scale has been understudied to date. The third gap is really trying to understand temporal change at broader scales and how the extent that ecosystems and ecological properties change in a synchronous way. And when we have synchronous change, where ecosystems are changing in the same way, it helps us be able to infer mechanisms. So synchrony is a really important characteristic of ecological properties. And then finally, variation in space and time of ecosystem properties. So things that ecosystem ecologists care about, matter and energy, as opposed to species, communities, and diversity. Species, community, and diversity is really important. I love animals. I have a dog. She's great. But I would argue that a lot of ecology has focused on biotic processes, particularly when talking about studying scales of variation, and we haven't studied as much the ecosystem matter, such as nutrients and energy. So these are the gaps that we felt like a program, a broad scale program studying lake nutrients could help fill. All right, so why does understanding ecosystem properties matter? Most ecologists are interested in biotic properties. Well, I would argue that increasingly ecologists are being asked to make some predictions and being asked to understand how ecosystems play a role in larger global processes. And so let's think about carbon cycling for one. Increasingly, aquatic ecosystems are recognized to contribute to global carbon cycles. And in fact, in L&O Letters, we're just about ready to publish a whole special issue on the role of carbon in inland, the role of inland waters in carbon cycling. And so, for example, to be able to say what inland waters are doing to the global carbon cycle, we need to be able to extrapolate from well-studied systems to less studied systems within a region. We also need to be able to scale up. Can we, from the well-studied system, say in Michigan, can we scale up to the region, and then how do we scale up to the globe? And then we also need to be able to forecast. If we can characterize what the regional carbon cycling is, can we say what it's going to be in 50 years from now? So studying ecosystem properties at these macro scales, I think, will help us be able to do these three important things that we should be doing as ecologists. Okay, so how should we go about doing this? Well, now is a really good time to be studying ecosystem processes at broad scales. Increasingly, we have bigger and bigger data sets available to us that are not only at broad spatial extents, but also at fine spatial grains. So we're able to study detailed processes across broad spatial extents. You think just 20 years ago, the computing power, we were limited by both storage space and computing power to be able to really do this at broad scales. But there's a lot of data out there that is allowing us to do these sorts of studies. Now, data is one thing, but we also have to have the concepts and the theory to drive the research that we're doing at this broad scale. So some have argued, well, macroecology and biogeography have been doing this for a long time. And they have, except, and the macroecologists would agree, that they're looking at large-scale ecological patterns of species and communities. And they have less to say about ecosystem properties. So macrosystems ecology is a relatively recent subfield that is emerging, and it's the study of ecological phenomena, both biotic and ecosystem matter and energy, at regional to continental scales. And so the research that we've done has been in the context of macrosystems ecology, which I want to just talk about and introduce a little bit. So macrosystems ecology, it represents sort of a new perspective. We don't think it needs its own journal and it needs its own professional society. Rather, we just think ecologists should broaden how they're viewing ecological systems to include this regional and continental scale. Macrosystems ecology has strong foundations in these well-studied areas of ecology, including landscape ecology that focuses on scale, spatial pattern and process and heterogeneity, hierarchy theory, coupled human natural systems, and of course macroecology and biogeography. So it borrows heavily from the concepts and theories from these existing subdisciplines. I'd like to acknowledge an effort that I worked on a couple years ago with a great group of colleagues, one of them sitting in the audience here. We got together several years ago, those of us who were funded on the National Science Foundation Macrosystems Biology program, to help try to define what macrosystems ecology is. So we did a special issue on the topic, and then Jim Heffernan and I co-led the lead paper that was tasked with defining what macrosystems ecology is. And so this is the framework we came up with. As I said before, ecologists have been studying global scales pretty well as local scales. But macrosystems ecology recognizes that there's also this intermediate scale at the range of thousands, hundreds to thousands of kilometers. And what macrosystems ecology, this framework recognizes that ecological systems are hierarchical, again, drawing heavily from hierarchy theory, and that there's interactions, important interactions across these scales. And we've characterized 4 types of interactions, and we laid this as a framework, as a way to study both local and global processes for a variety of ecological questions. All right, so we've taken this framework to conduct macrosystems ecology research on lakes, as I said. So we built a database called Lagos to conduct this research. And the database includes in situ samples for lake nutrients, productivity, and clarity, as well as the ecological context of lakes and the regions that they're nested in. So as Craig said, this is my study population. And as an ecologist, I've never before been able to say, I know exactly how many entities are in my population. There's 51,000. And that's really cool because you can start playing games when you know what every entity is in your population. So for these 51,000 lakes, we have all sorts of GIS characteristics of them. And you can see these lakes are nested within regions. The red boundaries are the regions we use to study these lakes and that have been found to be important boundaries for understanding these lakes. So, all right, so our goal in this research has been to understand spatial and temporal variation in lake ecosystem properties at macro scales and to understand what's controlling that variation. And to do that, there's 4 questions that really had to be answered before we could start building this understanding. The first is we might as well take advantage of research that's been done at the local scale. We know a lot about what drives lakes at local scales. So can we apply these local scaled concepts to lake nutrients at broader scale? So that's been an important theme of our research. The second theme is even trying to understand what the spatial structure of those 51,000 lakes are. How much do lakes vary across space? And what is the spatial structure of not only the lake properties, but also the factors that control those properties? Third is temporal variation. So you might argue, well, we've been studying lakes and ecosystems through time, through well-studied long-term monitoring programs, such as at the Great Lakes. But I would argue even well-studied systems we don't entirely understand. And so when you then try to extrapolate to, well, how are 50,000 lakes changing? There are some questions about temporal variation at broad scales we don't know, such as how have lake ecosystem properties changed in the last 2 to 3 decades? And are there spatial and temporal patterns of that change that would help us be able to extrapolate to unstudied lakes? And then finally, we want to put space and time together, as Stommel did 40, 50 years ago. And we want to ask, what is the relative amount of spatial and temporal variation? And by knowing the relative amounts of the variation, can that help us develop a better understanding of what's going on with the variation at macro scales? So our research has been designed to address these 4 questions. And so first, I'd like to talk about the great Lagos team that built Lagos. We spent 7 years to build it. We thought it would take 2, and it took 7. It's been fun. It involved an interdisciplinary team of aquatic ecologists, GIS experts, database experts, statisticians, and computer scientists. And it definitely was a collaborative effort. So the ecosystem properties that we compiled into Lagos were lake nutrients and measures of primary productivity. When we started this about 2010, we recognized that state agencies are responsible for monitoring lakes to meet the needs of the Clean Water Act. And yet those state data sets are often designed to deal with state issues, but not broad scale issues. And so nobody had put all of those data sets together before to ask some basic research questions. So we thought it was a really great opportunity to build a macrosystems ecology database from existing government data sources. And we also included citizen science monitoring programs, federal survey programs, and some university researchers. We also, based on the rich literature of what controls lake nutrients, we also have an idea of what might control them. So based on that research, we compiled all the GIS data sets that we thought would contribute to explaining lake nutrients, such as hydrology, lake watersheds and their connections, geology, and land use and atmospheric deposition. So we compiled all of this data together to create the database. So from the very beginning, the Macrosystems program at NSF was very much a proponent of sharing data sets, and so they required that we shared it at the end of the project, and we were very happy to do that, and it's been a fair amount of work to get it in an accessible form, but that has a lot of advantages because it forced us to document the database throughout. And it's freely available if you're interested in ever using it. So I also would like to acknowledge all the technical expertise that was needed to create this database, expertise that I do not have. So it's, again, it was a pretty major undertaking, and lots of people worked very hard to pull this off. Okay, this is just a glimpse of data that's in Lagos. And what I like about this GIF is it just shows when samples were taken, and the color scheme shows the density of samples. And you can see upper Midwest and the Northeast, we have a little more data than other states. It's also interesting, some states, started sampling well into, or as far back as the 70s, but most of our data start kicking in the 90s and 2000s. So this shows us where the samples are and the relative amount of the data. To give you an idea of the complete data set, the gray dots are every lake in our population, and the red dots are dots where we have some in situ measure. So you can see it's not evenly distributed, so that is something we have to factor in our statistical models, but it's a pretty good distribution across the study area. Okay, so I'd like to first start with the first question, which is trying to apply ecosystem scale concepts at macro scales. And I'd like to start with the phosphorus loading concept and how we might apply this to understand nutrients at broad scale. So the phosphorus loading concept, I don't think I have to go into too much detail here. It's phosphorus concentrations in lakes are a function of nutrient sources, their transport, which influence the loads, which themselves are influenced by internal processing, such as retention. So we can apply this in single lakes. We can apply this in many lakes. Phosphorus loads are very hard to measure, so often we infer phosphorus loads from measures of nutrient sources and transports. But we want to apply this in a macrosystems framework, so we also have to recognize that even multiple lakes, groups of lakes, are nested within regions, and regions have characteristics. And we think these regional characteristics will influence how the phosphorus loading concept ideas are expressed. So we applied this to the Lagos data set, the 4-digit hydrologic units, which is a watershed-based region, is our measure of region, and lakes are nested within these. We used hierarchical linear models, which recognizes this nested structure, and this shows the median phosphorus of lakes in the Lagos data set. We have 6,000 lakes with TP. And so we fit these models to try to see if the phosphorus loading concept would apply. And the boxes that are red are the specific variables in those categories that turned out to predict phosphorus across these broad scales. So we came up with variables that make a lot of sense, like land use, lake depth, residence time. And we also learned that there are some regional variables, nitrogen deposition and groundwater index, that also help to explain the regional expression of this. Now, you might be saying, well, what does nitrogen deposition have to do with phosphorus? And the simple answer is, I have no idea. We are still figuring out how to infer the mechanisms at these regional scales. My guess is, and I'll talk about this in a bit, it has to do with the spatial structure of where nitrogen deposition has changed in the last 30 years. But again, it gives us an idea of where to start looking. But it also made us think, well, would this work for nitrogen? And in fact, it does. And as you might expect, different predictor variables are going to predict nitrogen relative to phosphorus. And these are the variables that were important to predict for for nitrogen. Nitrogen deposition itself, runoff, land use cover, and lake depth. So again, this type of relatively simple modeling exercise shows that we can apply ecosystem scale models to explain macroscales. And importantly, both local and regional predictors were important to explain at the macro scale. You can't just look at the local scale. Okay, so the second question, and this kind of research definitely got us thinking about, well, what is the underlying spatial structure of all of these different variables? And does measuring that spatial structure, will that help us learn more about explaining variation at broad scales? So this work was led by a former postdoc, J.F. Lapierre, in which he asked specifically what is the spatial structure of lake ecosystem properties and their drivers. So what we were interested in doing is basically calculating the spatial autocorrelation of all of these variables. So you can do that by just doing a simple Moran's I, but a better way to do it is by calculating Moran eigenvector maps to quantify the spatial structure. And I'm just going to show you these. What you do is you create 1,000 maps with different amounts of spatial structure. You can see map A has very strong spatial structure, and then map F at the bottom has very weak spatial structure. And these maps you can then correlate with your variables to get an idea of what the spatial structure is of the underlying variables. So, for example, if we just plot out mean temperature that is at every lake in the data set, this is the mean temperature, it's air temperature. But you can see that has very strong spatial structure versus lake depth. And lake depth has relatively weak spatial structure, which means within a region you can get any lake depth. And so knowing which region a lake is in isn't going to help you predict lake depth. But what these MEMs allow us to do is actually quantify this spatial structure. Okay, so we quantified the MEMs, and then we calculated just simple correlations among pairs of variables with different spatial structure. Okay, so bear with me while I explain this figure. What we wanted to know is if variables with similar spatial structure had higher correlations with each other. And this gives us a little idea of whether predictor variables need to have the same spatial structure. So the variables plotted in blue have weak spatial structure. And what we're plotting is, let's say we're taking lake depth, and we're going to plot lake depth against every variable that either has very low spatial structure, which would be on the left, or has very high spatial structure, which would be a variable on the right. And these are the relationships we expect. Okay, so we took the group of variables that were weakly structured, we coded them as blue, and what this shows is that weakly structured variables were highly correlated with other weakly structured variables. Those are the dots in the circle. So it sort of matched our expectations, at least for weakly structured variables. And a great example here is lake depth. It has hardly any spatial structure. Then we looked at the strongly structured variables like mean temperature, and we correlated mean temperature to all the other variables along this spatial structure gradient. And what it shows is that strongly structured variables are correlated, highly correlated with other strongly structured. So it supports that prediction. And then finally, intermediate structured variables, the highest correlations are found with other intermediate structured variables. And so water clarity or Secchi depth is a great example of an intermediate structured variable. And so to give you a little more information of how these variables cluster, we found that lake morphometry, so like lake perimeter, lake area, lake depth, have very little weak spatial structure. Lake nutrients and productivity have intermediate spatial structure, and climate and land use have broad spatial structure. So this really helps us think about what we might expect as far as what predicts lake nutrients. So to sum up, we found the highest correlations were between variables with similar spatial structure, which matches a lot of early work published in the 70s and 80s from biotic systems. Having similar spatial structure is a necessary condition for strong correlations, but not a sufficient one. So you notice that there were a lot of high correlations in the zones where they should be, but there also were low correlations. So what it can tell us is the maximum, but it can't tell us the actual correlation. And then finally, importantly, lake nutrients and productivity are intermediate in spatial structure, which kind of makes sense when you think about the results from our first model that said both regional and local variables like lake depth predict lake nutrients. So you might expect the spatial structure of lake nutrients then to be intermediate between those two drivers. So that, at least the two themes of our research align, which is good. I was happy when that happened. Okay, so that's space. And we've spent definitely more time thinking about space, but fortunately we have members on our team who also think about time. And so given this data set, again, that went back to most about the 80s or 90s, we thought this was a neat opportunity to say, well, how has water quality changed since the implementation of the Clean Water Act? And are there spatial and temporal patterns to it? So Sam Oliver took the lead on this effort. And we used another hierarchical linear model to ask this question of how lake properties have changed. But we use a slightly different form of the model. It's an unconditional 3-level model where we have random effects for region and lake. So what that means is that we can model the lake change through time, but we can also model sort of the regional signal of trend. Hierarchical models are great for a lot of reasons. In addition to the hierarchy, they also accommodate uneven sampling designs across space and time, which is this data set is unfortunately uneven. Okay, so what are some of the results? So what these dots show is the change in total phosphorus and total nitrogen, and the percent change ranges from -5 to 5. So that's percent change per year. So relatively small change. And when we fit the model to the entire population for phosphorus, there's been no change. So yes, there's different colored dots, but statistically across all the lakes, there hasn't been a change. And that's pretty important to acknowledge. And then for nitrogen, though, we did notice a significant change of a decrease in total nitrogen of 1.1% per year across the lakes for which we had nitrogen, which is interesting and sort of makes sense when you think about that changes in nitrogen deposition have been pretty remarkable in the last 20 to 30 years. So we were able to detect that in our data set. Now, fitting a global model is fine and interesting, but odds are there's a lot of interesting within region dynamics. And we did find that, for example, for phosphorus on the left, 7% of the lakes did increase, and 9% of the lakes decreased in phosphorus. So this means, although we're not seeing any major dramatic change, some lakes are increasing, some lakes are decreasing, likely due to complex local factors that might be interacting with regional factors. And actually, we were able to explain some of these changes, but not well. And then for TN, most of the lakes decreased, 11% of them, but 2% of them did increase. So it's nice to be able to characterize the results both at the population scale as well as the lake scale. So despite management efforts to control eutrophication and global change, we don't see overwhelming support for the idea that all lakes are getting more and more eutrophic. And that's an important message. Oops. Okay. So then the second part of this question is, okay, so that's just looking at overall change. What about how are the lakes changing, and is there some spatial clumping to how lakes might be changing? So Noah Lottig led this effort where we used Secchi disk measurements because there's a lot more data on Secchi. So we actually were able to compile 600 long-term time series for Secchi in the range of 22 to 25. Some will argue whether that's long-term, but that's pretty long. And so we can ask the question of how are our lakes changing across the last 25 years. And here we used a k-means clustering approach, a computer science approach, and we looked at correlations among pairs of lakes to see if they're changing synchronously. So this is our data set here, and it just shows median Secchi for those 600 lakes. So when we're thinking about patterns of change, our previous model, the hierarchical linear model, assumes a linear change. But we also know from a lot of long-term studies that there are other kinds of change that can happen through time, nonlinear, threshold, or even anomalous type change. And so with this study, we wanted to sort of test this idea that, well, is our linear models really fitting what's going on in lakes, and also address the issue of whether they're changing synchronously. So this is these are the clusters of the 600 lake time series. We've clustered them into 8 different clusters. And so the black line is the fitted line with uncertainty around it. And then the gray lines are the individual lake. And what you can see is that we have one of the clusters that you could say, yep, that's kind of linear. The other clusters are either some nonlinear or possibly a threshold type response, at least on average. And so this result suggests, you know what, we may need to be thinking about approaches that can factor in nonlinear dynamics. So next, what we wanted to do is say, okay, well, for example, cluster 2, it looks like there's a linear increase in clarity, which means water quality is getting better. So we might think, is that happening all in one region? You might expect that it is. So there's cluster 2 again. The red dots are all the lakes that are in cluster 2. And so the answer to that, are they all in one region, is no, they're not. They're spread pretty far and wide. And we were actually quite surprised by this result. We expected a lot more spatial synchrony, which means lakes that are nearby experiencing similar land use, similar regional nitrogen deposition, similar climate, would respond similarly. So we have some spatial clustering, but not much. And this was a pretty surprising result. And to look at that more more quantitatively, what we did is we took every possible lake pair, correlated them with each other, and then plotted it along a distance gradient. If there was a lot of spatial clustering, then you would have expected very high correlations on the left side of the graph and lower correlations as you got farther away. And what you can see is, yes, there's some very high correlations at small, short distances, but there really isn't a strong relationship that would suggest a lot of spatial synchrony in temporal change in lakes. Okay, so what have we learned about time in lake nutrients? Well, there's been relatively little change in nutrients across these broad scales and across the last 20 years. And although there were synchronous patterns, so lakes did change similarly, they were not strongly spatially structured. So this is in contrast to the average condition of lakes, which have some spatial structure to it. The temporal signal does not seem to have that spatial structure. Okay, the final theme that I'd like to talk about is trying to pull it all together and looking at space and time together, and what can we say about the relative amounts of spatial and temporal variation in lake nutrients. So just to give you an idea of the variation that we have in lakes, so I've already shown you some maps of median values of phosphorus. That's the map on the left. And not surprisingly, the Corn Belt has a lot of phosphorus. The northern lakes do not. But what's interesting is looking at the variation estimated as the coefficient of variation. It doesn't seem like there's a lot of spatial pattern in the variation. And what we also wanted to try to understand is better understand this kind of temporal variation that's going on, regardless of whether it's linear or nonlinear. Okay, so the approach we took here is a Bayesian hierarchical variance component model with random effects. And the key part here is the variance components. It allows us to break down the different types of variation. We also then could add predictors in the model to predict the different components of the variation. So the components that we're looking at, we quantified, were regional spatial variation, local spatial variation, which would be within a region, synchronous temporal variation, and then ephemeral. And just to give you a quick idea of what these are, so spatial regional, spatial local probably makes sense. If you have strongly spatial signal, then lakes within a region are going to be very similar. If you have a lot of local variation, then lakes within a region are going to be very different. And then for these looking at time series, a synchronous time series is where the ecosystem response is going to change similarly through time. Ephemeral is basically each of the lakes is doing their own thing. So that some other local factor is causing the change, and it is not happening synchronously. So we can look at these maps like this, but the variance component model allows us to actually quantify these types of patterns, which is what we did. And so you might expect, so in calculating variance components, you're basically taking all of the variation in the data set as 100%, and then you're breaking it down into the different components. So what's plotted here, each of these add up to 100. I've just split them out, spatial, temporal, and residual. This is what we might expect. We might expect a bunch of spatial variation split between local and regional, somehow temporal split between synchronous and ephemeral, and then residual. And this is what we got. For 8,000 lakes that we had roughly 25 years of data. As perhaps is not too surprising given the results I've already shared, is there was a lot more spatial variation than temporal. The spatial variation is split pretty evenly between the local and the regional, which means, again, at these broad scales, we cannot ignore what's going on with the region. And for the temporal, at these broad scales, at least looking at annual variation, there's not a whole lot of it, and a lot more of it is ephemeral than synchronous. So, and of course, there's also 20% of variation that's residual that includes error, measurement error, components that we haven't measured. So this kind of accounting can help us better figure out what's going on when we're trying to predict at broad scales. Okay, so then what about, can we predict the variation at the different scales? So this shows the estimated effect sizes for the different components. So the spatial regional, as I said before, for phosphorus is related to a groundwater index and a change in nitrogen deposition. We found spatial local was related to local variables that we would have expected from the literature. So it confirms what we know. And we weren't able to model the temporal or the synchronous temporal variation. And that's not surprising. Remember, it was really small, that variation. So we think we just don't have enough variation to explain. So we weren't surprised by that. We were disappointed, but not surprised. And then we can calculate how much of the variance we can explain at each of these levels. And what's really cool is at the regional level, we can explain 88% of that regional level just with these two variables, which for these kind of models is really high. For the local variation, we can explain about half, and that matches what's in the literature. And again, we can't explain anything about the temporal. Okay, so to sum up what we know about space and time variation, spatial variation is divided almost equally between regional and local. Ephemeral temporal variation far exceeds synchronous. And this helps us figure out how to better understand temporal dynamics that maybe a regional approach isn't going to help. And then finally, spatial variation is 5 times temporal variation at these scales. So all of these conclusions are based on the spatial extent of this study. Okay, so what have we learned about scale and variation in ecology? We think we've learned something. We provide empirical support for the fact that the dominant predictors of ecosystem properties differ by scale. We have very concrete evidence that shows that's the case. On average, interannual average state of ecosystems will vary little through time. And we think this is in line with a lot of recent interest in regime shifts work and looking at temporal change in well-studied systems, that it does take large and sustained perturbations to move systems far from their mean. And our work would sort of lend support to that maybe these systems that are experiencing pretty big change are more rare than we might think. As spatial extent increases, we should expect less synchronous temporal variation. And this also makes sense when you sit and think about it, because if you think about the spatial structure of ecosystems, if lake nutrients are intermediate, then when you're looking at a broad spatial extent, you wouldn't expect synchrony because that spatial structure starts breaking down. So maybe we shouldn't have expected to see synchrony. And then finally, at macro scales, spatial variation is likely to always exceed temporal variation. And I would also argue that we would propose these as hypotheses to be tested in other systems and for other variables. Okay, so we've also learned some very specific answers about lake nutrients at macro scales. So we were happy to see that we could apply the phosphorus loading concept at macro scales. Lake nutrients and productivity have an intermediate spatial structure, which is not something we knew prior to this, and I think will really help us understand lake nutrients at broad scales. Across the last 20 years, there's been relatively small changes in the average state of nutrients and productivity. There may be less synchronous dynamics in lakes than would be expected. There have been a lot of work on synchrony in lakes from regional studies, and that's why we thought we would see more synchrony. So this issue still needs to be worked out. And then finally, spatial variation and productivity is roughly split between local and regional and is about 5 times larger than temporal. And we also tested this for nitrogen, Secchi, and chlorophyll, and these results came out pretty similar. Okay, so what's the next steps? Well, we're gluttons for punishment, and we want to spend another 7 years building another database, which is exactly what we're doing. And we're expanding to the entire US scale. Because again, we want to expand the scope and look at even broader scales to see if what we've learned at the subcontinent scale applies to the continental scale. And finally, we think this kind of approach will help us integrate knowledge across scales by focusing on the relationships and controls of variation, but also quantifying that variation with methods like the variance component modeling and considering the spatial structure of systems to better be able to inform our efforts to extrapolate, scale up, and forecast, which increasingly we need to be able to do in the face of global change. Thank you.
Questions for Pat, and I guess we have to speak to the microphone. I got a couple on the top of my head.
I think there's one there.
So I think that the comparison of space versus time and the strength of their effects is really interesting. And but we tend to have a lot more data across space than we do through time.
Right.
And so how much of that comparison do you think is related to sampling effect, and how do we get at testing that hypothesis?
Right. Yeah. So we've actually talked about that a lot in our group. And whether our results are biased by the fact that we've got better spatial data than temporal. I would say looking at 600 time series with the Secchi analysis, is pretty good. But I would argue, so again, our conclusions are based on the annual scale. One scale we couldn't say anything about is the subannual, so within summer dynamics. And if we had data, that's the scale that I really wish we had, that we could look at within annual variation and how that changes our view of temporal variation. To me, that's a really big unknown. I think we can get at the issue of, are we biased because we have less temporal data? I think we could get that with the simulation study. And we talked about doing that for at least 2 of these papers, but it turned out to be beyond the scope of both papers. But I think that's an important next step. Would be, at the moment, we're not going to just create new data. So I think it would have to be a simulation. And I think we've got enough data and understanding that maybe we could simulate it to see if we are unnecessarily biased by our data set. It's a great question.
Hey, how's it going? I'm not sure if you said this in the beginning, but where were you getting all the data in your data set? Is that government data? Is that labs? What's all that?
So it's mostly state agency data sets. So, when I first moved to Michigan 20 plus years ago, I started working with the state agency, and state agencies have to monitor their inland lakes, and they have to submit a report to EPA every 2 years. And I'm like, well, that's kind of neat. So I started compiling the data that Michigan samples. And I was, for Michigan, I think I had 700 lakes. I'm like, wow, this is an amazing data set. I started out at the state level. And then I realized, well, every state should have an equally good data set. And so what's interesting, and they did, there's a lot of state to state variation. But what's interesting is a lot of the state agencies have partnered with citizen scientists. And so now a lot of the data are collected by citizen scientists, sent to state labs. So it's sort of a consortium of state and citizen programs that collect a lot of this water quality. Then we also got some data from university researchers, although not much, because they're not as good with data management. So and tribal. There are several tribes in the area that have regulatory rights to manage their water, so they also sample their water. So we got some data from tribes, and then some federal, large surveys. Also, was a lot of acid rain monitoring in the east, and we grabbed those data sets. So basically anything. So we were like a group of about 12 limnologists, and we just said, okay, what's all the data we can think of? And that's what we came up with. There was a lot of data just laying around.
Mm-hmm. And so I guess for me, my concern would be, as with any scaling up process, is how do you think do you feel comfortable really comparing the data? Do you feel like it's sampled the same way across the board? Do you feel good about quality control, that kind of thing? Yeah?
Yeah, that's a really good question. So we've written numerous papers about our methods, just documenting how we quality controlled. The quality control, that's what took the 7 years, to be honest. It was all the quality control. And the other reason I thought this was going to be a good idea is because, think about it, the EPA has some fairly standard methods for measuring things like nutrients and chlorophyll. And the states have to write a QAP, a quality assurance plan, and submit it to the EPA. So to be honest, they're doing a lot more careful QA/QC than we as university researchers do. I mean, yes, I'm careful when I go out and sample, but they've got to document a lot more than we do when you think about it. And so because they're government data sets, we had a lot of documentation, so we could say, yes, this method applies and it works. And actually, most of the times we've tried to only take data that use the same method. But yeah, that's the biggest challenge in building these data sets. Yeah. Yep.
We have questions?
Thank you. This is a really interesting big data analytic project. I got two quick questions. So for the next 7 years, when you expand to the rest of US and continental, what's going to be your strategy? Do you going to small region every year, or are you going to scatter around and then adding few more each year?
Right.
And do you expect any new insight or difference for the rest of the new region?
Right.
The second question is, given this comprehensive database for this region, and you get all the relationship with spatial and temporal scale, let's say we have a known lake in Europe or in Africa, and we know very little or nothing about it.
Right.
How could a researcher get more insight or learn anything from this existing database?
Right. Okay, I'm going to answer that question first because it's been right in my mind, and we're trying to tackle that right now. So what we're going to try to do is take the Lagos data set. We're going to say, okay, let's say we have data on the eastern US, but not the Midwest. We're going to build our models. We're going to try to build the best predictive models we can. And then we're going to validate the model with the data in the Midwest. Now, my argument would be that model's probably not going to do very well. But what we're going to do is a series of validation experiments to figure out what are good ways to extrapolate and what are bad ways. So for example, is it enough to just have 5 lakes in a region, but spread across the entire study extent? Or do you want one region sampled really well, and then you can apply that knowledge to another region that you have no data on? So we're going to start by doing those experiments with the 17 states. Then once we have our US states, once we have 48 states, we can continue those experiments, and we'll need to do that at the extent of the US because there's arid regions, right? There's climate and geophysical settings in the West that we don't have in the Northeast and the Midwest. So again, all of this depends on the spatial extent. So I think once we expand into arid regions, we'll be able to see, could we extrapolate, say, to, South America or to Africa? A lot of it depends on what the inputs for our driver variables are. let's say we can find another biome that matches the biome in the US exactly, then we might be able to extrapolate. But to me, this is what we're going to do with that data set of the 51,000 lakes, right? Because we know every lake. We can start playing these games. So yeah, and actually the extrapolation and the scaling up is a big focus of this grant, the US grant. And then the first question is, how are we going to select lakes to add in the new? Well, again, it just depends on available data. We're going to, again, go back to state agencies and just get the data that they've collected. But I think your question is getting at, could we make recommendations to monitoring agencies about where they should be sampling? And I think the short answer is yes, we definitely could. And that's where I think the spatial and temporal variance component can help us. If all we want to do is get a rough idea of the regional water quality, say in a state, then you should sample through space and you can kind of ignore time. That's what I would recommend. You can say, look, you're going to get that plus or minus 20%. But if you really care about time, I don't know what the answer is, because again, like you said, we maybe don't have enough temporal data to say what's the best way to get at how systems are changing. That we just have less knowledge to ground our expectations on.
We have questions that, Kathy. Whoops, sorry. Where's the mic?
Great presentation, really enjoyed it. Also studied at the University of Wisconsin Center for Limnology. So
yay, yeah, go Badgers.
We speak the same language. At any rate, I wanted to ask if integrated into your research, if management is being integrated, or is this basically just looking at variation? And where does the management component work in? Maybe it's beyond the scope of your research, but I was just curious.
Yeah, no, that's a great question. And I would say one of the articles, the Sam Oliver's article where she looked at the change through time, the biggest criticism from one of the reviewers was, there's all sorts of management going on. You should add information about the management. And yes, that would be great. The challenge is we just don't have that information. I would say that would be, part of the reason we made Lagos open access is to make it available now. If somebody has a way to collect all the information about what management has happened on these lakes, they can build that data set and just click it into Lagos and ask all sorts of really cool questions. I would love to do that down the road. Another interesting layer to add to this is policy, right? There's land use policy at the local and regional scale. There's state policy about how they deal with nutrients and nutrient criteria. And that's just been beyond the scope of our work. But I'm hoping to either collaborate with people to do it down the road, or I hope somebody else takes that on, because it would really add an important component. And I think it might help explain that additional local variation. There's still about half of the local variation we can't explain. And I think, of course, things like food web dynamics and within season dynamics matter, but I wouldn't be surprised if management's going to be an important player at that scale as well.
Patricia, there's one comment. Regarding the use of the 4-digit HUC, it might have been better to use underlying geology, basement versus glacial till versus Paleozoic.
Yeah, that's a great question. We've also had numerous debates about what's the best way to delineate the region. I would say we selected HUC 4, again, because it's watershed based. So we wanted lakes that might be connected to be in the same region. We thought that connectivity would matter. And so a watershed based region will give us that. But you're entirely right. There could be, for nutrients, basing the regions on soil and superficial geology might be a better way. And those are, some of our research is designed to, and for this next round, we're going to try to build different kinds of regions and test which ones might be better. We've done some of that work, And Huck 4 has turned out to be better than other ways to carve up the land. But there's still more work that needs to be done in that area, definitely.
Sorry, I'm calling on you here without giving you the mic.
Yeah, nice presentation. I was curious, you mentioned acid rain data. So I happened to be recently looking at some of those data, and there have been big changes in precipitation chemistry over the time period of your study. So I was kind of interested in seeing in your presentation if some of that would come out, because there might be, large continental scale temporal trends as a result of that. There were like decreasing nitrate deposition, increasing ammonium deposition, increasing pH of precipitation.
Yeah.
And I was curious if you were specifically looking for effects from those changes, and was it surprising that you didn't see much in terms of temporal trends?
Yeah, well, remember, so we're just looking at phosphorus, chlorophyll, water clarity, and nitrogen. And in those sort of more water quality variables, I think maybe they're not as directly connected to acid rain and deposition as maybe alkalinity, pH, conductivity might be. So in the next round, we are adding those variables to the data set, and I think we'd be able to pick up more of a signal there. But something's going on with nitrogen deposition, because it often pops out, whether it's nitrogen deposition in 90 or the change in nitrogen deposition. But that there's something regional going on there that we haven't been able to tease apart.
Yeah, so you noticed a correlation between nitrogen deposition and phosphorus loading in your model?
Yeah.
So could that be related to changes in precipitation chemistry and phosphorus mobility or something like that?
Yeah, it very well could be. And yeah, we just haven't gone into that level of detail to try to tease that apart. There's also really interesting, we have a fair amount of nitrate data in our database. If you're interested, go download it. I've wanted to get someone on our team to look at it, particularly people who know nitrate, like Emily Stanley, but they've been too busy. There are some really interesting patterns that are going on with nitrate that I think would be more directly tied to nitrogen deposition, and we just haven't had the time to do it. But yeah, I think there's something going on there, and we just haven't fully explored it. So yeah, let me know if you find anything.
The data are available for anybody.
They are available for
them.
download for a small price. No, I'm joking. They're free.
All right. I think we'll cut it off there, and Pat will be around for a little bit if anybody wants to chat with her or ask more questions. And let's thank her again for an excellent presentation.
Muted.